So in this lab, we'll be using fruit flies, and the reason being is fruit flies have been used as a model organism in genetics for about 100 years. They have a short life cycle and are easy to maintain in the laboratory, and the genetics is very well understood. The DNA sequence for the Drosophila genome was published in 2000. Drosophila has only 4 pairs of chromosomes, and genome contains 180 MB of DNA and about 14,000 genes. So the purpose of this experiment is to evaluate the effects of ethanol in flies which have altered genes such as ADH plus and ADH minus. So there will be there will be one control group to which the flies will be compared to. The fruit flies will also have, the fruit flies with the mutated gene for the enzyme alcohol dehydrogenase will be our main test subjects. So our group hypothesis for this lab is that we predicted that the fruit flies which were exposed to alcohol, which had the mutated gene for breaking down alcohol, which was ADH minus, would die off by the end of the experiment. Then we also predicted that the flies which do not have the mutated gene, which are ADH plus flies, would have no problem surviving in any environment they encounter, whether it being alcoholic or in water. So this table depicts the data that we collected from the flies which had ADH plus and ADH minus gene, which were put into environment of water. As you can see on the left hand side, we have the time, which are in intervals of which start out as 0, 15, 30, and an hour to hour 25 minutes, and finally a 24-hour period. For each of these intervals, we collected the data of how many flies were still present and moving were alive during these times, and recorded the data under the survivors. So as you can see, the ADH plus frequency in water had 100%. It was 100%. No flies passed away, and in ADH minus, the frequency was 100% till the 24-hour period, which it dropped down to 83% because one fly had died. This table shows the flies which were put into an alcoholic environment. As you can see, the ADH flies had no deaths in this test. It's 100% frequency survivors. However, the ADH minus flies started off at 100%, and at the 30-minute period dropped to 66% because one fly died. Then again, at the 24-hour period, we have 0% alive because all flies were dead. There was no survivors. So the observations that we observed during this lab, and for ADH plus flies and alcohol, at time 0, all the flies were sticking towards the sides of the wall, walking around. At 15 minutes, the flies were very active, remaining closer to the top of the container. 30 minutes, all the flies were active, walking around the test tubes, and at 1 hour, flies were actively moving towards the top of the tube. Then at the 24-hour period, we had 4 flies which were alive and active, and showed that there was no flies which were dead. So observations we observed in ADH minus in alcohol was at time 0, 2 flies were active on the side and 1 was remaining on the cotton ball. At 15 minutes, 2 flies were active near the top and 1 remained on the cotton ball, inactive. 30 minutes, 1 fly has died, laying on its back at the bottom. And at 1 hour, both the remaining flies are active. At the 24-hour period, all flies have passed away, with no survivors. Observations for ADH plus in water. At time 0, both flies were clinging onto the side of the wall. 15 minutes, both flies were active at the top. 30 minutes, 1 fly is on the cotton ball, 1 fly is active on the wall. At 1 hour, 1 fly was active moving around near the cotton ball. At 24-hour period, both flies were very active, still moving around. Observations for ADH minus in water. Time 0, 5 flies are sticking onto the top, while one is on the cotton ball. At 15 minutes, 6 flies were active. 2 of them were on the cotton ball and near the top. At 30 minutes, 5 active flies near the top, and 1 is on the cotton ball. At 1 hour, 4 active flies near the top. 2 remain on the near the cotton ball. At the 24-hour period, 1 fly has died. The rest 5 flies are active. In this chart, you see the flies at the interval 0. So all the flies the class started out with in this picture. So this graph shows us the class data of the flies which were alive at the 24 interval. So as you can see, there are only 3 bars. This is because the alcohol, the ADH minus flies and alcohol, they all died in every group's results. So the results that we got from our data was in ADH minus flies, which were put in water, we experienced 1 death at the 24-hour period. Whereas the ADH minus flies in alcohol, we experienced 1 death at the 30-minute interval, and then at the 24-hour interval, we had 0 flies, which meant all the remaining 2 flies have also passed away. So we had 2 sets of flies. Our control group of flies had a normal functioning gene for the enzyme ADH. These flies are able to produce alcohol dehydrogenase, which breaks down alcohol. The other set of flies were genetically modified to have a mutated ADH gene. These flies are not able to break down alcohol because they're missing the enzyme ADH. The set of flies that had a mutated gene for ADH and exposed alcohol had the most deaths because they were unable to break down alcohol. The control group of flies exposed to alcohol all survived, which was expected since they have the gene to catalyze the oxidation of alcohol. So in our experiment, we did not witness any deaths from ADH plus group. This is because they have the gene to break down alcohol, which helped them to survive the alcoholic or water environment. However, none of the ADH minus flies survived the alcoholic environment because they did not have the gene to break down alcohol.